A single-celled organism is a master of all trades. It breathes, eats, moves, and reproduces entirely on its own. But that biological independence has a strict size limit. To build a trillion-cell redwood tree or a human being, a single cell simply cannot handle the physical workload of keeping an entire massive structure alive. To bypass that size limit, evolution developed a solution called cell differentiation. Instead of remaining generic clones, newly divided cells physically change their shape, size, and internal structure to take on highly specialized single-focus jobs. When these specialized cells group together by the millions to perform the exact same job, they form what we call a tissue. Tissues are the hidden architectural units constructing every living thing you can see. As highlighted in this textbook definition, a massive organism isn't one solid object. It is an intricate matrix of these microscopic, highly coordinated worker teams, dividing the labor of survival so the entire structure can thrive. We do not have to guess how this microscopic architecture operates. The evidence is hiding in plain sight, and you can physically prove it. Using a few basic investigative steps, examining a sprouting onion, a cut flower, and a simple glass slide, we can map out the hidden engineering systems driving both plants and animals. The most accurate way to understand complex life is to physically look under the microscope and watch how it divides its labor step by step. We start by finding the exact epicenter of a plant's growth. If you take a common onion bulb and suspend it in a glass of water for several days, it will begin to sprout roots. By isolating those roots and measuring their length every 24 hours, a distinct pattern emerges. Roots do not stretch uniformly from the base. All physical lengthening happens exclusively at the tips. This diagram of a plant shoot illustrates why. The isolated growth zone is meristematic tissue, cells capable of rapid division pushing the plant forward. Panning down, we see the wake. Maturing cells form permanent tissue, losing the ability to divide, locking into fixed structural shapes. A plant does not grow everywhere at once. It uses highly concentrated teams of dividing meristems to push outward, leaving behind a hardened permanent scaffolding to support its weight. To see how that permanent scaffolding functions, we look at this experiment: a white petunia stem placed in a beaker of red-colored water. After a few hours, we cut a transverse cross-section of the stem to examine it under the microscope. The red dye acts as a biological tracker. To the naked eye, a stem looks solid, but the dye proves a directional flow system exists, engineered inside the plant. Looking at this textbook inference, specific tubular cells absorb the red dye. This is the xylem, a tissue acting as a rigid, one-way pipeline designed to conduct water upward. Next to the xylem is its partner tissue, detailed in this diagram, the phloem. While xylem cells are mostly dead and hollowed out for water flow, the phloem is living tissue composed of sieve tubes and companion cells, built to transport food across the plant. A plant survives strictly through this dual plumbing system: dead, rigid pipes pulling water up, perfectly paired with a flexible, living highway distributing nutrients wherever they are needed. Animal biology requires an entirely different engineering strategy. We can map it by creating a monolayer blood smear on a glass slide. Because most animal cells are translucent, microscopists apply a Giemsa stain to the smear. This chemical reaction forces invisible components, like granules inside white blood cells, to pop with vivid color. This table directly compares the liquid tissues now visible under the lens. You have blood, a red fluid matrix designed to transport oxygen and nutrients, contrasting with lymph, a colorless fluid matrix dedicated entirely to your immune defense system. Now compare those flowing liquids to the supportive connective tissues shown in this chart. Bone relies on a hard, calcified matrix to form a rigid skeleton, while cartilage uses a flexible, protein-based matrix to absorb shock at the joints. Plants can rely on stiff, unchanging walls, but animals are highly mobile. Their connective tissues span extreme physical states, from flowing liquids to solid rock, to support constant movement. A mobile machine built out of free-flowing liquids, heavy bones, and contracting muscles would instantly collapse without a centralized control system issuing coordinated orders. That control system is nervous tissue. Looking at this diagram of a single neuron, you can trace the path of command. Branching dendrites at the top perceive incoming sensory signals, and the long cylindrical axon propagates those signals outward to trigger muscle movement. The neuron is the master biological switch. By firing electrical impulses across this network, it turns a heavy collection of bone and blood into an interactive, self-aware organism. Stepping back from the microscope, the engineering choices we've observed highlight the ultimate evolutionary divide between a stationary plant and a mobile animal. This comparison table summarizes the split perfectly. Plant tissues are largely dead at maturity, using minimal energy to provide rigid, stationary support. Animal tissues are living, highly complex, and burn massive amounts of energy to sustain constant movement and hunting. In biology, form strictly follows its function. The microscopic shape of an organism's tissues is a direct, logical mirror of its survival strategy. Everyday concepts like flesh or wood are biological illusions. They are highly organized, microscopic labor forces, perfectly engineered for their environment. The next time you look at the sharp veins of a leaf, or feel the pulse beating in your wrist, you know exactly what is happening beneath the surface. You are feeling the invisible, specialized tissue systems dividing the labor, keeping the machine alive.